Good afternoon. I'm Dr. Brian Shapiro, a marketing segment manager at ATCC. Thank you for joining us for the latest installments in the ATCC Excellence in Research webinar series. It's entitled Bioluminescent Insights, Reporter Cell Lines and Checkpoint Inhibitor Therapeutic Development, presented by Drs. Utsav Sharma, Haiyun Chong, and Kevin Tayo. Dr. Sharma is a product manager, while Drs. Chong and Tayo are scientists in immuno-oncology research and development at ATCC. In this presentation, our speakers will present new reporter cancer cell lines that have high endogenous expression of immune checkpoint molecules. They will then discuss the application of these novel reporter cell lines in gamma interferon stimulation assays and immune cell co-culture assays and share insights on how these cell lines can be adopted into checkpoint reporter assay workflows. If you have any questions for our speakers, please use the chat function available through the webinar program. All questions will be answered as time allows at the end of the presentation. The recorded webinar presentation will be archived on the ATCC website, www.atcc.org. So with that, I'd like to welcome our first speaker, Utsav Sharma.
Before we begin, a brief overview about ATCC. Founded in 1925, ATCC is a nonprofit organization headquartered in Manassas, Virginia, with an R&D and services center in Gaithersburg, Maryland. We are one of the largest and most diverse biological materials and information resource for cell biology, often referred to as the gold standard when it comes to authenticated cell materials. We are an innovative R&D company that is focused on developing new and advanced cell models and tools to accelerate oncology, immuno-oncology, drug discovery, and screening. With that, I'd like to briefly take the opportunity to showcase our oncology portfolio of products broken down by application here. At ATCC, we offer classical cell products in the form of human and animal cell lines, certified reference materials, as well as other cell culture media and reagents. For biomarker discovery, we have matched tumor and normal cell lines, quantitative cell line DNA, as well as cancer cell panels. We support tumor biology research by a collection of reporter-labeled cell lines and cell lines broken down by gene mutations. For drug screening applications, we have a multitude of primary cells from various tissue types at low passages, CRISPR-engineered isogenic cell lines, as well as a robust cell immortalization program. For immuno-oncology applications, we have our classical portfolio of primary immune cells and cell lines, hybridomas for antibody manufacture, THP1 monocyte cell line in various reporter formats, as well as our newest offering of CAR-T target and checkpoint reporter cells that you'll be hearing about today in today's webinar. Finally, we have a growing collection of patient-derived models offered in collaboration with the Human Cancer Models Initiative, or the HCMI, in the form of organoids, adherent and suspension cells, as well as conditionally reprogrammed cells that we have companion growth kits and exhaustive cell culture guides for easy adoption into immuno-oncology platforms. In today's webinar, our scientists will provide a brief overview of the field of immuno-oncology, share data on protein profiling of the ATCC cell lines, and provide insights into application data that we've generated with the checkpoint reporter cell lines. Followed by will be a functional comparisons of the modified and parental cell line. At the end of the webinar, there'll be a Q&A session. Please use the chat function to drop in any questions you may have during our session, and they'll be answered at the end of the webinar during the Q&A session. With that, I'll hand over to Dr. Haiyun Chang.
Thank you, Utsav, for the introduction. Good afternoon, everyone. My name is Haiyun Chang, and I'm a scientist at ATCC. Today, I'm going to introduce you to ATCC's newly launched immune checkpoint reporter cell lines and explain why and how we developed these cell lines. Immune checkpoint molecules are the markers on the cell surface that communicate self to the immune cells. When T cells find these markers on the surface of another cell, they recognize the cell expressing these markers as self and do not attack. Cancer cells use this to their advantage and increase the expression of these markers to evade immune detection. Scientists have found and studied quite a few of these immune checkpoint molecules, and examples of them are shown in the picture on the left. Of these molecules, the reporter cell lines that we will be introducing today endogenously express high levels of PD-L1, CD155, and B7H3. These markers are important in cancer research because we can develop therapeutic antibodies against them and block the interaction to initiate T cell mediated immune responses in the tumor microenvironment. The images on the right show you an example of how these immune checkpoint inhibitors work. Therapeutic antibodies against PD-L1 and PD-1 bind each target molecule on the cancer cell and T cell respectively and prevent the immune checkpoint recognition. This leads to the activation of the T cell to begin the killing of the cancer cell. The history of these immune checkpoint inhibitors is short, but they have become a significant breakthrough in the field of cancer immunotherapy. I will talk about the history of immune checkpoint inhibitors briefly in the next slide. The first immune checkpoint molecule was discovered in 1987 and named cytotoxic T lymphocyte antigen number 4, or CTLA4. However, the function of this molecule was not clearly understood until 1995 when Dr. James Allison and his team identified it as a crucial immune checkpoint molecule with great potential as a future anti-cancer therapy target. The first CTLA4 blocking antibody was immediately developed and tested on animals a year later in 1996. Soon after, programmed cell death 1, or PD-1, was identified as another immune checkpoint receptor expressed on T cells. The first checkpoint inhibitor approved by the FDA was ipilimumab, a CTLA antibody, in 2011 for the therapy of advanced melanoma. Subsequently, two PD-1 antibodies, pembrolizumab and nivolumab, obtained FDA approval in 2014. Since then, the FDA has approved 9 checkpoint inhibitors that target the CTLA4, PD-1, PD-L1, and LAG-3 pathways for the treatment of more than a dozen major cancer types. In 2018, Drs. James Allison and Tasuku Honjo were awarded Nobel Prize in Physiology and Medicine for their pioneering work on checkpoint molecules CTLA4 and PD-1 respectively. Even after about 2 decades of explosive growth, however, considerable challenges remain in development of immune checkpoint inhibitors. One of the major challenges is the intrinsic complexity of cancer immunology study models. Currently in the market, there are in vitro artificial systems available comprised of cancer cells that are engineered to overexpress certain immune checkpoint molecules and non-cytotoxic T cells that are engineered to express T cell activation signaling reporter genes. These in vitro systems are relatively easy to use and the results are generally reproducible, but since these cells are artificially engineered, the subsequent intracellular events following immune checkpoint inhibitor-induced immune activation are missing from the system, which is crucial in maintaining the physiological relevance of the system. Another frequently used model type is syngeneic or humanized mouse models. These are classic immunology models that enable us to understand the immunological mechanisms as a whole and test potential therapeutics. However, the interspecies disparity between the mouse and the human immune system is inevitable. The fully human ex vivo assay models that utilize human T cells and human cancer cells solve this issue. These are typically co-culture assay models of primary immune cells and patient samples that maintain physiological relevance, unlike the artificial models. However, the intrinsic donor variability issue makes it especially difficult to obtain reproducible results using these assay models. So what is the solution? A physiologically relevant assay system that can generate highly reproducible data would be the solution. So at ATCC, we have developed the immune checkpoint reporter cell lines that endogenously express high levels of immune checkpoint molecules. These reporter cell lines provide the physiological relevance that is more relevant and more predictive to the in vivo events, unlike the currently available artificial in vitro assay models. We have also achieved high reproducibility with these reporter cell lines by utilizing immortalized cell lines that eliminate the donor variability issue that is intrinsic to the donor-dependent human ex vivo assay systems. And by inserting luciferase reporter gene, the cell lines allow convenient data collection and enable reliable measurement of the potency and stability of immune checkpoint inhibitors. And through the next set of slides, I will explain more in detail how we develop the immune checkpoint reporter cell lines. For physiological relevance, we first wanted to find the cell lines that naturally express high levels of immune checkpoint proteins. We performed protein profiling by flow cytometry of over 50 cancer cell lines of various origin available at ATCC. The cell lines were profiled for both inhibitory and co-stimulatory immune checkpoint molecule ligands in the presence and absence of interferon gamma, a cytotoxic cytokine released by the immune cells. A heat map was generated based on the median fluorescence intensity, and each column was color-coded separately to avoid cross-comparison among different antibodies. The data here show which cell lines endogenously express which immune checkpoint molecules, and we selected our candidate reporter cell lines based on this information. As highlighted in the table, the three immune checkpoint reporter cell lines that we're going to introduce to you today were developed from MG63, a breast cancer cell line with high expression of CD155, a TIGIT ligand, NCI-H1650, a lung cancer cell line, which expresses a high level of B7H3, and HCC827, another lung cancer cell line that endogenously expresses a high level of PDL1. After identifying the cell lines that endogenously express high levels of checkpoint molecules, we devised a reporter assay system that utilizes a luciferase reporter gene for an easy, rapid, and sensitive quantification of the target gene expression. The lentigas Luke2P plasmid contains the luciferase reporter gene under the control of gamma interferon activation site or GAS response element. So when the interferon gamma is released from the immune cells and activates the JAK-STAT signaling pathway, it would induce the transcription of the luciferase reporter gene. After the viral transduction of the plasmid into the selected cell lines, the cells were then enriched by puromycin selection, and single cells were isolated by automatic cell sorting. The single cell clones were expanded and evaluated by interferon gamma stimulation. The clone that yielded the brightest luminescence upon interferon gamma stimulation was selected to be developed into the immune checkpoint reporter cell line. Here, this picture shows the mechanism of action for the immune checkpoint reporter cell lines. Normally, an immune checkpoint ligand expressed on the surface of a cancer cell, such as PDL1, would bind a complementary immune checkpoint receptor on the surface of a T cell and inhibit the activation of the T cell. when the binding is disrupted by an immune checkpoint inhibitor, such as a PD-1, PD-L1 blocking antibody, the T cell gets activated and releases various cytokines, including interferon gamma. The released interferon gamma is then recognized by the interferon gamma receptor on the surface of the cancer cell, and this leads to the activation of the intracellular JAK-STAT signaling pathway. The JAK-STAT signaling pathway activation induces the interferon gamma activation vector binding to Gas promoter, and in our system, this promotes the Gas-induced transcription of the luciferase gene. The series of events produces easily detectable bioluminescent signal that enables reliable measurement of the potency and the stability of the immune checkpoint inhibitor. Now, Dr. Kevin Tayo will come up and present the data we have generated using these immune checkpoint reporter cell lines.
Thank you, Hyun, and good day, everyone. My name is Kevin Tayo, a scientist at ATCC. Before I dive into our functional data, I would like to take some time to talk about our work in ensuring that these reporter cell lines maintain physiological relevance to their original parental counterparts. Here, we wanted to ensure that the physical properties of the unmodified parental cells are preserved in our Gas-LUC2 modified reporter cell lines following viral transduction. Here, we show representative microscopy images of some of our reporter cell candidates. At the top, we show representative images of parental HCC 827, MG63, and H1650 cell lines, as well as their Gas-LUC2 modified counterparts shown below. As you can see, the morphology of the modified cell reporters exhibit no discernible physical differences compared to their parental counterparts. Next, we compare the growth rates and cell doubling times between parental and Gas-LUC2 modified cells. For this study, we incubated cells for 5 to 7 days and assessed changes in the cell numbers daily. These graphs illustrate the daily increase in cell number for each cell line during their designated incubation time. Using this data, we were able to calculate the doubling time for each cell and compare differences in growth rates between parental and modified cells. Here, we show that all three reporter cell lines exhibit similar growth rates relative to their parental cell analogs, all showing changes in growth rates well below 20%. Additional studies were conducted to ensure that the high target expression in each monoclonal Gas-LUC2 cell line was comparable to their parental equivalents. Here, flow cytometry analysis was performed to compare the expression of PD-L1, CD155, and B7H3 in HCC 827, MG63, and H1650 cell lines, respectively. These graphs illustrate the expression of the respective target, shown in red, as well as an isotype control, shown in blue. Expression of the parental cells are shown on top, whereas their Gas-LUC2 modified cell counterparts are shown below. For all three reporter cell lines, the target expression was shown to be comparable to their unmodified parental cell lines. Additional flow cytometry studies were conducted to ensure that the target expression in modified cells would be maintained following prolonged cell culturing. Here, we compare the respective target expression in low passage number cells, shown on top, to cells cultured above 30 population doubling levels, or PDL, shown at the bottom. Here, we show that high expression is maintained in all three cells, even after prolonged culturing. Next, I want to switch gears and discuss some of the application data we generated, showcasing some of the functional studies of these modified cell lines. So following the Gas-LUC2 plasma transfection of our candidate cell lines, we wanted to examine which cell line for each target would provide the highest change in luminescent signaling from the interferon gamma stimulation. Here, we show the results of a series of interferon gamma stimulation assays of candidate immune checkpoint reporter cell lines for PD-L1, CD155, and B7H3. All cell lines here showed increased luminescent signaling upon stimulation with the increasing concentrations of interferon gamma. The cell lines that yielded the highest fold change relative to untreated controls were selected for the next set of experiments. For PDL1 reporter cell lines, HCC827 gas loop 2 cells consistently showed the highest fold change in luminescent signaling following stimulation with various interferon gamma concentrations. Similarly, MG63 and H1650 cells were selected for CD155 and B7H3 respectively. After clonal selection, the 3 cell lines were once again tested with cytokine stimulation. All 3 cell lines demonstrated high sensitivity to 24-hour interferon gamma administration, especially the HCC827 gas loop 2 cells, which exhibited an average of 50-fold increase in luminescence relative to untreated controls following a 0.01 nanograms per mil interferon gamma administration. Increased luminescence was observed in all cell lines with increasing interferon gamma administration, with 1,000 nanograms per mil interferon gamma administration resulting in a range of 125 to 250-fold increase in luminescence compared to untreated controls. Additional interferon gamma stimulation tests were conducted on reporter cells with high PDLs to ensure that each reporter cell line maintained high luminescent signaling from stimulation even after extensive culturing periods. Here, we show that even after 30 population doubling levels, all 3 reporter cell lines maintain high luminescent signaling. Following the interferon gamma stimulation assays, the signaling activation was also assessed using conditioned media from primary human T cells. Here, the 3 cell lines were treated for 24 hours with conditioned media from either non-activated or anti-CD2, CD3, CD28 bead activated primary human CD8 cytotoxic T cells, followed by luminescence detection. The conditioned media from the activated CD8 cytotoxic T cells increased luminescence values by over 100-fold in all 3 cell lines, relative to media taken from non-activated T cells. Next, the ability of the cell lines to serve as a checkpoint inhibitor reporter was evaluated by primary human CD8 cytotoxic T cell co-culture studies. Here, monoclonal HCC827 gas loop 2 cells were co-cultured with different ratios of CD8 cytotoxic T cells, as well as under different co-culturing times. The cells were also administered with various concentrations of either a PDL1 monoclonal antibody or an isotype control IgG1. Increasing the PDL1 antibody concentrations in the co-culture models resulted in a dose-dependent increase in the luciferase expression, exhibiting a 3- and 5-fold increase of luminescence signaling relative to untreated controls under the conditions of a 24 and 2-hour co-culture incubation period with a 1:1 and 1:10 ratios of HCC827 gas loop 2 to CD8 T cells, respectively. In contrast, increasing isotype control IgG1 concentrations failed to induce any luminescent signaling from HCC827 gas loop 2 cells relative to untreated controls. Co-culture studies were also conducted on MG63 gas loop 2 reporter cells. This time, cells were cultured with CD4 helper T cells at 24 and 48-hour incubation periods. Here, cells were administered various concentrations of either a TIGIT monoclonal antibody or an isotype control. Again, we observed that increasing TIGIT antibody concentrations in the co-culture models results in a dose-dependent increasing of luminescence intensity. Here, luminescent signaling was 3- and 6-fold larger than untreated controls for a 24 and 48-hour incubation times, respectively. Again, there was no observed increase in luminescence intensity from increasing concentrations of the isotype control from treated cells relative to untreated controls. Finally, co-culture studies were conducted on H1650 cells using natural killer cells at a 2:1 ratio. Incubation times for these co-culture studies were conducted at either 24 or 48 hours. Here, co-culture models were administered with increased concentrations of either a B7H3 antibody or IgG controls. Again, increasing the administered concentrations of B7H3 antibody in the co-culture models results in a dose-dependent increasing of luminescence intensity, with a 2- and 3-fold increase luminescence signaling relative to untreated controls at 24 and 48-hour incubation times, respectively. Yet again, there was no observed increase in luminescence intensity from increasing the concentrations of the isotype control relative to untreated controls. Now, let's summarize our findings. The recent success in the use of immune checkpoint inhibitors in the treatment of cancer shows enormous promise. However, new tools and models in the field of immuno-oncology are desperately needed. To serve this urgent need, ATCC conducted an extensive protein profiling of our substantial catalog of cancer cell lines, T cell lines, and primary T cells to assess for high expression of selected immune checkpoint molecules and their ligands. Based on this protein profiling data, we have generated and developed several immune checkpoint reporter cancer cell lines for use in the convenient detection of immune checkpoint blockade-induced signaling. Here, we showcase 3 cell lines that provide stable and robust luciferase signaling produced by relevant stimulation, enabling reliable measurement of the potency and stability of immune checkpoint inhibitors. The endogenous high expression of PD-L1, CD155, and B7H3 and HCC827 gas loop 2, MG63 gas loop 2, and H1615 gas loop 2 reporter cell lines, respectively, delivers physiological relevance to the immune checkpoint assay. For more information on this topic, please visit us at atcc.org/immuno-oncology.
Thank you, Hayun and Kevin, for the insightful presentation. In just a few minutes, we will begin our Q&A session. At this time, please use the chat function available through the webinar program to submit any questions. The recorded webinar presentation will be available on demand on the ATCC website at www.atcc.org.
All righty, well, thank you for the excellent presentation. In just a few moments, we will begin our Q&A session. Please remember to use the chat function available through the webinar program to submit your questions. And I can see that we've already had a bunch of questions come in, so let's go ahead and jump in. So this first question, what is the passage limit of the immune checkpoint reporter tumor cell line?
Okay, so I'll get this question. So we have tested and confirmed the robust expression of the luciferase in all 3 cell lines at over 30 population doubling levels.
Okay, good. Here's another one for Hayun. By the way, Kevin had to step out of the room for a moment, so these are mostly going to be going to you, Hayun. Do you have to use CD8 positive T cells for the co-culture?
So no, it is not necessary to use just CD8 positive T cells for co-culture. Any immune cells that express the checkpoint molecules will be able to be used. We have used CD8 positive T cells, CD4 positive T cells, and NK cells as well. The luciferase expression is in response to the presence of interferon gamma in these cells, so any immune cells that produce interferon gamma can be used.
All right, sounds good. Now, Hayun, is it necessary to use activated T cells for co-culture?
So that's a really good question. Activated T cells release high level of interferon gamma, so it will allow the immune checkpoint reporter tumor cells to emit strong lucifer signal. And this is useful if you want to use T-cell conditioned media to elicit the luminescent signal. However, if you're testing an immune checkpoint inhibitor that activates T cells by blocking the immune checkpoint binding, then you may want to start with the T cells that are not activated at a lower activation level. so that you can have the you can keep the background signal low, and those immune checkpoint inhibitors will activate the T cells and thus elicit a stronger luciferase signal from the cancer cells.
Nice, nice. Now, Hayoon, could you talk a little bit about the co-culture system that you use? So how did you actually co-culture the checkpoint reporter cells with T cells? Did you use CD8 positive T cells or CD4 positive T cells? And do you have any recommendations for optimizing co-culture conditions?
So we used both CD8 positive T cells and CD4 positive T cells with different cell lines. So it really depends on your need. But one thing that you want to keep in mind is that we are measuring luminescence, luciferase expression. So if the cells are killed, if the cancer cells are killed, then you won't be able to see the luminescence from these luciferase expressing cells. And CD8 positive cells are cytotoxic T cells. So once activated, these cells will kill the tumor reporter cells. So you will need to optimize for your need, the type of cells that you're using, and you can also optimize the time of the co-culture, and you can also optimize the ratio between the T cells and the cancer cells. So for example, I have done like 1:1, 1:2, 1:5, 1:10, like different ratios of cancer cells to immune cells. And also for the time points, you can do 6 hours, 24 hours, 48 hours, and you can actually see that when you're using CD8 positive T cells, as the cytotoxic T cells kill the cancer cells, the luminescence level actually goes down.
Okay, great answer, great answer. Now, going back to the original question about activating T cells, how does ATCC activate the T cells?
So we use commercially available anti-biotin B particles that are loaded with biotin-related antibodies against human CD2, CD3, and CD28 antibodies. So those are the commercially available T cell activating kits, and we use them regularly, like every 2 to 3 weeks, to keep the T cells activated.
Gotcha. Hey, I'm just jumping around a little bit here to make sure that we capture all the questions. I do have a question around licensing, and this might be one for Utsoff to cover, although I can grab it also. Are there any additional licensing requirements for the cell line?
Great question, Brian. Thank you for passing that on to me. There are some specific licensing requirements that are associated with these cell lines that we've showcased today. The full licensing and the material transfer agreement requirements, they can be found under the permits and restrictions section on our product page for the website. So when you access the ATCC website at www.atcc.org, you'd be able to look under the permits and restrictions section for this particular part. And if you need to specifically have a question around licensing, you can also reach out directly at salespermit@atcc.org.
Okay, great. All right, back to Hayoon for some more technical questions. So Hayoon, how does ATCC go about doing single-cell cloning? And were these cell lines developed that way?
So yes, so these cell lines are developed by single-cell cloning. We use an automated single-cell sorter for cell sorting.
Okay. Can you use these cell lines to support bispecific checkpoint inhibitor screening?
So that is a very good question. we have this, we have profiled the cell lines for the most common checkpoint molecule expression, including co-stimulatory molecules, and that data is available on the ATCC website. And you can find that some of these cell lines, they have more than one checkpoint molecules expressed, highly expressed on the surface. And you can choose those and use those for bispecific inhibitor screening. But we have not done any screening at ATCC, but this is something that you can probably work on your own. Look through the protein profiling data and see like what other checkpoint molecules are expressed on the surface of these cell lines.
Great, great. Now, can these cell lines be used for other checkpoints besides PD-L1, CD155, and TIGIT? And maybe another way of putting this is, are there any additional cell lines that ATCC may have for that have checkpoint luciferase expression as well?
So another good question. Yes. Yeah, so we do have other immune checkpoint reporter cell lines available right now. So for the ones that we have for the cancer cell lines, they express PD-L1, CD155, or B7-H3. And those are the ones that we tested and verified, but then they also express other like high levels of some other checkpoint molecules as well. So that is one thing. We do, and in addition to that, we do have other cell lines available. We have a T cell line with NSET-LIG2 that has a PD-1 as a biomarker. We also have 3 bioloid cell lines, 2 bioloid cell lines that's currently available, and 1 more that's coming soon. And those are those cells express high levels of SIRP alpha, cyclic-10, or PD-L1. So we have more than one. We have more than just the cancer cell lines. We have cancer cell T cell lines and bioloid cell lines, and we also have other checkpoint molecules on this on the surface of these cell lines as well.
Gotcha. Okay, a couple other questions have come in that I'm going to send Utsav's way. So Utsav, this first question might be out of the realm of this particular presentation, but you might be able to answer it anyways. So this group is interested in a couple of things. First of all, they have some substances that are killing MCF7 and MDA-MB231 cells. They're looking for mechanisms and are interested in reporter lines that might illuminate some of the different pathways of interest, one of them being IRE1 pathway, specifically involving BIP and CHOP proteins. Could you maybe expand on if there's ATCC has anything that might be able to help them out?
So very interesting question. Certainly outside the checkpoint realm of reporters, ATCC has developed MCF7 and MCF10A, which is a companion normal to MCF7s, and an MD-MB231 CRISPR-engineered cell line. These cell lines are unique. They belong to our EMT-MET reporter cell line portfolio. They express a fusion protein that's directly under the promoter that promotes vimentin. So they, when activated or constitutively express vimentin RFP as a fusion protein. Now, IRE1 and BIP and CHOP proteins, I believe, are the unfolded protein response proteins, or something to do with protein translocation, as much as I remember my pathways. So I'm not sure if you are specifically interested in looking at EMT, and if these proteins are probably going to be involved in EMT. If they are, then that would be a good reporter to study. But we don't have any specific reporters that have specifically engineered reporter constructs looking at that particular pathway.
Right. That is excellent feedback, by the way, that we can pass along to our product development R&D groups that you're interested in that. Okay, then jumping to the next question that I'm going to direct at Utsof. Can these cell lines be used in vivo, like in xenograft studies?
Great. Another great question. So all of these cell lines, specifically the tumor cell lines that we really dived into the data about today, the ATCC 827 gas loop 2 cell line, that's an adenocarcinoma. I believe if you inject about 500,000 to 1 million cells, you can get 7 days post-injection, pretty good tumor. The MG63 and the NCI H160 have a ton of resources available online that can show you tumor growth in vivo xenografts as well. So that's an osteosarcoma background for the MG63, and the NCI H160 is an adenocarcinoma as well. So all of the experiments that we highlighted in vitro here today can be done as in vivo xenografts. Our newer releases that Hayun alluded to earlier, that we also have coverage for the T cells and the myeloid cells in this compartment for the immune tumor microenvironment. Those come from lymphoblastic leukemia, AML, and lymphoma backgrounds. So those also can be used in xenograft studies in vivo, and you may be able to assess checkpoint inhibitor efficacy and potency in xenograft.
Nice, very nice. Hey, this next question I'm going to send back Hayun's way, and also we'll make this last call for questions as it's the last one that has come in. So, Hayun, can you comment on will the fold increase of signal increase even past 48 hours?
So that is a great question. It depends on what type of experiment setting that you're using. So if it's just the interferon gamma stimulation assay, or whether it's a co-culture assay. So co-culture assay, in the co-culture setting, the signals tend to come a little later. So with that, we do see it's not very common, but then we do see, and it's not like a big increase. We usually see the biggest signal increase at around like 24 hours or to 48 hours. But then we do sometimes see a little like delayed response with co-culture, depending on like how activated the T cells are. If it's just a simple interferon gamma stimulation assay, then the signal, the peak signal we usually see at around 6 to 8 hours, and they will decrease after that.
All right, excellent, excellent. Well, thank you, Hayun, Kevin, and Utsof for the excellent presentation, and thank you everyone for attending this webinar. If you would like to learn more, you can navigate to www.atcc.org/immuno-oncology. Additionally, we are going to be exhibiting at the American Association of Cancer Research annual meeting this year in sunny San Diego at the San Diego Convention Center. So if you're there, please stop by booth 1413 to learn more about these and other ATCC advanced models. Thank you again, everyone, for attending, and have a great day.
Thank you.
Thank you.